Yeah, hi everybody and good afternoon. And gives me great pleasure to introduce Bharat Rastogi. Bharat did his bachelor's from St. Stephen's in Delhi and a master's from UC Santa Barbara, where he worked on cloud-plant interactions. That was the short version of it. And then went on to do his PhD at Oregon State, which he'll be talking about. Bharat's a freshly minted PhD, something that I know that many ATREE students aspire to be any day now, and some people have finished, and for some people, of course, it's in the distance. But this is a good moment, right, when you've just finished and are moving on. So Bharat's going to be talking to us about his PhD work. And Bharat, I mean, the very first student to graduate from ATREE, PhD program, was also a Bharat. So your name has special resonance for the ATREE Academy. Yeah, with that, I'll hand over to Bharat. You guys can probably move a little bit closer. It's a small group. If when you see me slip away in a few minutes, it's not because I'm not interested in Bharat's work. I heard his talk on Thursday in IASE. So I've, yeah, with it. Thank you.
Hello, is this working? Yeah. Thanks, Karthik, for that nice introduction. And thanks everyone for coming to listen to me talk about my doctoral research. I hope to stop doing this soon and move on to other projects. Spend some time on this. So the title of the talk is on the screen: An atmospheric tracer sheds new light on terrestrial photosynthesis, and I'll talk about some field work that I'd done during my PhD in an old growth forest. So just a brief outline, just to set the context on global warming and the carbon cycle, how this obscure gas called carbonyl sulfide, which few people have really heard of or worked on. I hadn't heard of it before I started working on it. What the role of carbonyl sulfide is, and how we can measure and monitor mixing ratios of carbonyl sulfide in forest canopy airspaces to infer photosynthesis by estimating fluxes of carbonyl sulfide, and then compare productivity from OCS with that from other approaches and some overall conclusions. So just to start off very briefly, this is a plot of temperatures relative to sort of mid-industrial level, just showing that globally temperatures have increased. So we're looking at the red line here, which shows at least from 1950 to about 2018, 2020 onwards, the rate of increase of temperatures relative to the mid-19th century. And on this they've also plotted when these key summits had happened, and finally we've come to some sort of agreement in Paris, but already we've seen about 1.5 degrees almost of warming. This is primarily due to increasing CO2 levels in the atmosphere. So this is a reading I got from this is from Mauna Loa Observatory, which has kept the longest record of continuous measurements of CO2. And it's in this isolated place. It's in Hawaii on a mountaintop, this observatory. And they've been measuring since 1958. You can see that concentrations have sort of been building up ever since measurements began. And if you extend this far back into time, you'd see the rate of increase is much higher. In fact, we're now at CO2 levels that are highest in the last 800,000 years or so. That encompasses lots of glacial, interglacial cycles. So increasing CO2, warming temperatures, this is sort of well known by now. So just to look at quickly what happens to anthropogenic emissions. So we've been emitting CO2 primarily by burning fossil fuels and through land use change, which account for about 88 and 12% of our emissions. There are other emissions, but really these are the main sort of emissions, main anthropogenic emissions. And we can see that about half of our emissions stay in the atmosphere. So about 46% or 17 gigatons CO2, sphere is staying in the atmosphere, and the rest is sort of divided into ocean and land sinks. So the ocean accounts for about 8-9 gigatons of CO2 per year, but the land surface really is the biggest sink of carbon. Biggest sink of carbon from sort of helping to remove CO2 from the atmosphere. And there is some uncertainty here as well, about 6%, which are really sort of big numbers. This uncertainty is primarily comes from this estimate of land sink, which we are really not able to constrain. And I'll show this here on this slide, which is paper by Debbie Huntsinger, published recently in Nature Scientific Reports, which shows that models don't even get the sign. So if you're looking at these red dots across all these models, you can see that they don't even agree on the sign of the cumulative net land sink. So some models will say that the land surface overall has been a source. Most models will say that it's a sink, but really we don't have a good handle of what this number is. We don't really know what the cumulative net land sink has been. This has implications on climate feedbacks. So our ability to model global temperatures is somewhat quite dependent on how well we understand the land sink. And so there have been studies on this saying that in CMAP5 climate projections, there are uncertainties due to carbon cycle feedbacks. And this is that paper that I showed the figure from earlier, which says that the uncertainty in the response of terrestrial carbon sink to environmental drivers undermines the carbon climate feedback predictions. Really long title, but really gets to the issue. This is primarily because the land sink is composed of two opposing fluxes, or has to be derived from these two opposing fluxes, which are respiration and photosynthesis, or GPP. The problem here is that there are no direct measurements of GPP beyond the leaf level. So if there are some plant physiologists in the room, you probably had a lycor instrument that you can clamp onto a leaf or a shoot, and you can get net photosynthesis from that instrument pretty well. But extrapolating from that, going bigger than that, we really have no direct measurements. And that makes it difficult to resolve over large spatial scales. So what we do at the ecosystem scale, which is somewhat sort of zooming out from a single leaf to maybe a stand scale, is using this method called the Eddy covariance method, which I'm not sure if some of you are familiar with. But what it allows us to do is essentially measure the net exchange of CO2. It's called NEE, the net ecosystem exchange, over a plane. So in this case, this is a forest surface, and we have some eddy covariance instrumentation above this forest, which is telling us what the net exchange of CO2 is across this surface. And then we have to partition that into GPP, which is the land sink from those previous slides, and ecosystem respiration. We don't really have direct measurements of each, so we have to use some sort of algorithms or some sort of model that allows us to understand respiration and GPP independently, and then sort of partition NEE metrics. This is where carbonyl sulfide comes in. Carbonyl sulfide is chemically very similar to CO2. So this is CO2 on the left and OCS on the right. Instead of a second oxygen atom, it has a sulfur atom. So it's chemically very similar. Most abundant reduced sulfur gas in the atmosphere, with the mean atmospheric concentrations of 500 parts per trillion. And parts per trillion is about, so it's about 1 million times less than CO2. So it's very minute amounts. I'm just wondering how I'm doing on time. That's the P45, right? Okay, sorry. Thanks. Am I going too fast? Slow down. So carbonyl sulfide is this sort of like a chubby cousin of CO2, some of us like to put it, with a second heavier sulfur atom instead of an oxygen, but much smaller mean atmospheric concentration. And when people started measuring this at some of these long-term measurement sites installed by NOAA, which is the National Oceanic and Atmospheric Administration, They saw that they were, across the growing season. So this is sort of looking at 1 year, different months. And these are mixing ratios measured at these northern hemispheric sites, terrestrial sites over land. People are measuring carbonyl sulfide. And they saw that this sort of pattern resembled, what you would see for CO2, which is basically uptake during the growing season in the northern hemisphere. And that the drawdown, the magnitude of drawdown was highly related to that of CO2. So you could really see that at sites where there was a larger drawdown in COS, it corresponded to a larger drawdown in CO2, right? So there's some sort of plant influence which was seen across these large this network of sites, and this is because it's primarily consumed in the biosphere by carbonic anhydrase. And carbonic anhydrase is a ubiquitous enzyme, but it's also present in inside the leaves of plants, and it's used in hydrolyzing CO2. So the primary step in photosynthesis involves hydrolysis of CO2, and the same enzyme can catalyzes hydrolysis of carbonyl sulfide. So they have a very similar uptake pathway. So this is sort of a schematic from a paper by Joe Berry in 2013, which sort of showed us that synthesizing from a couple of different studies, that the uptake pathway of COS or OCS, it's the same gas, and CO2 is very similar. So they follow the same uptake pathway from the atmosphere inside the leaf. And this is shown in this, where this is a cross section of a leaf surface, and you could see that the uptake is modulated by the boundary layer and then stomatal conductance, diffusion through the mesophyll, and then some sort of biochemical activity. Does this sort of make sense? I don't think so. Rubisco is also involved in. No, and also you have to remember that the mean atmospheric concentrations are much smaller, so it doesn't really affect. It doesn't really interact with CO2, but the uptake pathway is very similar. The only difference is that there are no emissions from photosynthesizing plants, so it's not involved in the respiration cycle at all. So CO2, like we were saying, is always fixed and respired by the plant or the leaf surface. Carbonyl sulfide is a one-way flux, so it's only going in from the atmosphere into the leaf. And so people have started to study this at larger spatial scales. A few years ago, from a few days of measurements, these guys across a network of sites in Israel found that you can infer ecosystem photosynthesis from measurements of carbonyl sulfides. And so there have been a few papers on it very recently, some very fascinating research. But the idea basically is that if you can measure both COS and CO2 at the same time, you can get a direct estimate of photosynthesis that you can't get just from measuring CO2 fluxes, because you don't have to do the partitioning, essentially. And so in this paper, they sort of lay out this was sort of a validation exercise where you could infer canopy level stomatal conductance, which is sort of mediating CO2 uptake by plants using carbonyl sulfide. So the idea is if we measure both, if we can get a good estimate of stomatal conductance by carbonyl sulfide, we can get a direct estimates of GPP. However, there are very few measurements, and a lot of observations remain unexplained. A lot of these studies that I showed previously focused only on a few days of measurement, and a lot of ecosystems have not been studied. So it's been sort of few days of measurements here, few days of measurements there, and there are no measurements from old growth forests. This was sort of setting the stage for my research. So I'm just going to talk a little bit about So we are at Wind River, which is in this in the northwestern United States. Zooming in a little bit, we're over here. So this is the site, Wind River, which is about 100 miles, 160 kilometers inland from the Pacific Ocean. The climate is sort of temperate oceanic, or basically a little cooler than Mediterranean ecosystems, which really means that most of the rainfall occurs in the winter months. So summers are warm and dry, and winters are cool and wet. So it doesn't get very cold. Of course, those are subjective, but but usually doesn't get very cold. It's very wet during the most of the year, except for the summers, which are sunny and clear and dry conditions usually. So ecosystem consists of old growth forest. This is a picture of a tower coming through the forest where we mounted our instruments. I'll talk a little bit about this in a second. But most of the trees here are, there are only 2 species, very, very boring. But there are Douglas fir and western hemlock trees. Most of the tallest trees are Douglas fir trees, and the highest trees are about 60 meters tall. So they're really big old trees. One thing to note about these forests is that they maintain leaf area quite deep into the canopy. And this is sort of been important for my research, but also it's one way that old growth forests differ from, at least in the Northwest, differ from second growth forests, where there's a very diverse distribution of leaf area. In addition to these trees, there's a vibrant understory, several shrubs, and grasses are here found here. Also thick carpets of moss. So if you walk on the forest floor, you're basically just walking on green carpet. There's no real exposed soil surfaces here. Very green, lush sort of understory. In terms of what we measured, so we were at this site. This is a figure I showed earlier, which has this sensor, which is measuring CO2 fluxes and also temperature, energy, all sorts of environmental variables we're measuring. This is a power sensor, so getting at incoming light. So yeah, we're about 10 meters above the forest canopy, looking at all of this movement of air and stuff in it. But we also installed a profiling system. So this is what our system looks like. So this is that tower, which is about 70 meters tall. And we had 4 profiles. Later on, we added another one. But for most of the time, we had these 4 profiles at 4 heights. So we had measurements at 70, 60 meters, which gets at that above canopy sort of airspace. And then we also measured stuff in the understory at 10 and 1 meters. So the way this is done briefly is that we sort of pull in air simultaneously from all these lines. They go through some sort of a T, and from which they're fed into the analyzer, which is a spectroscopy. It does spectroscopy basically. It's in situ spectroscopy, and so we can get the concentrations of these gases instantly. And we do some sort of calibration every now and then or regularly actually, every 2 and 12 hours we perform calibrations to see if our instrument is drifting or not. And I can talk more about the system, profiling system later on if anyone wants to. But essentially what we do is we collect very high frequency measurements of gas at these 4 different heights, and then we calculate half-hourly and hourly averages to try and analyze and interpret the data. Yeah, again, this is just showing that picture. So we have sort of lines running through the forest. So there's some line which is at the top of the tower at 60, and then other lines sort of just coming out above from the base of the tower into the forest. So just very quickly into methods, we sort of we measure these profiles, and then we can get at fluxes by looking at gradients. So try and sort of estimate what the conductance is, what the aerodynamic conductance is, or conductance can be sort of, you can think of it as the opposite of resistance. So what the resistance to flow is between these 2 heights and what the gradient is between these 2. And then additionally, we can look at this thing called change in storage flux. So not everything in the ecosystem can be measured at the top of the canopy. There's some still air that we need to account for. And this sort of stuff will make a little more sense as I go when I explain the results. But in the end, we can get at the net ecosystem exchange by summing up the turbulent flux, which is FOCS here, obtained from flux gradient, and the change in storage flux. So it's the net ecosystem exchange is the sum of the turbulent flux and the storage flux, essentially. Just looking at this, we started collecting data in midsummer 2014. These are average diurnal cycles for about 40-45 days of measurements, and done some other measurements, but this work was focusing on this time period. So you can look at it, sort of nice diurnal cycles of temperatures. Get about 10 degrees C at the top of the tower, and then about 25-26 Celsius mean average temperatures during the summer. Pronounced cycles of humidity. These dotted lines are sunrise and sunset. And I've plotted corresponding soil moisture. So you can see soil moisture is continuously declined from when we started measuring to when we finished at all these heights. This means that there are no influxes of water into the system. So there are no rain events during this time period. So soil moisture starts out, say, at 40 centimeters, about 25%, or and then it goes down to about 20. So continuously drawn down, mostly by plants through transpiration. But there are no influxes of water in this ecosystem. Just jumping straight into the results, these are again average diurnal cycles. And what we see here is CO2 on the left, carbonyl sulfide on the right. Colors signify different heights. What we see is that the patterns for CO2, for instance, the average diurnal cycle for CO2 is very characteristic of vegetated surfaces. So you have some sort of increase because of respiration that suddenly tanks as plants start fixing carbon. So if you measure CO2 over a forest surface, you'll see this diurnal cycle, which reveals the influence of photosynthesis on the air around it. Drawdown of CO2 due to photosynthesis. When we saw carbonyl sulfide, it looked exactly the opposite, which made no sense to us at the time. And we were amongst the first people measuring this stuff, so we sent the instrument back, saying that this can't be right. You guys haven't gotten it correctly, and then they sent it back to us like, no, the instrument's working fine. So we sort of started thinking more about it then. But essentially, what you can see is that the average mean diurnal cycle is exactly the opposite. So there's some drawdown during the night, and then suddenly there's a spike that starts around shortly, a few hours after sunrise, and concentrations rise, and then they start declining again around 3 p.m., 4 p.m. The one thing that is important to note here is that concentrations of carbonyl sulfide are always higher above the canopy than lower down in the canopy. So the blue line here is 70 meters, purple line is 1 meter. So you can see there's a persistent gradient which is going from the canopy to the forest floor. So above, so if you were to put the forest in a box, which is kind of what we're doing here, see that the concentration above the box is higher than lower in the box. So we knew there was something different happening, but that the forest was taking up carbonyl sulfide, as evidenced by this gradient from top to bottom. So to put our study in regional context, we ran this model called high split. I don't know if any of you are familiar with this, but essentially it's a very simple model that can project trajectories backward and forward. So essentially used by so what it does essentially is that you have a source, you have a site. So I put my latitude and longitude, and then I say that, okay, what the air that is over this site, I ask the model to tell me where it's coming from. So it does, it uses reanalysis data, other data sets, and then tracks air masses. You can also run it in the forward mode, which is essentially where this model is used most of the time. For instance, if you have a plume of pollution or something, you can say, okay, if I release a plume of pollution over this factory or something, where is it going to go to? How is it going to disperse? But in this case, we're doing a backward trajectory, which tells us that all the air that we are seeing here at the site is coming in from the ocean. And what we know is that oceans are a major source of atmospheric carbonyl sulfide. So our hypothesis in this case was that what we were seeing here, this midday sort of emission, midday increase, was when the forest became coupled to the atmosphere, all this oceanic air rushed in, making the concentrations go up. We also have some measurements that I haven't shown. So there are some measurements from a mountaintop nearby, which also show much higher concentration. So they're not influenced by plants because you're at the top of a mountain. And so at mean atmospheric concentrations are higher, forest concentrations are lower during the daytime. All this air from the ocean rushes in, causes our concentrations to increase. But we still have a persistent gradient here. So we now need to use those other methods to estimate fluxes, which we did. So now on the left, I have fluxes of carbonyl sulfide, again, mean diurnal cycles. And on the right, I have fluxes of CO2. And you can see, so this is that, as I mentioned earlier, the net ecosystem exchange, which is the blue line, is the sum of change in storage, which is S, OCS, which is obtained by integrating along the entire profile, and the turbulent flux, which is estimated by that gradient at the top. And so SOCS, which sort of accounts for that entrainment signal, entrainment of oceanic air, shows that it captures that profile really well. So during midday, suddenly there's a spike as the ocean gets coupled to the atmosphere. All this air comes gushing in, and then it sort of dissipates during the day. The turbulent flux shows a complete opposite story. And in all of this, and the sign convention is unfortunately going to change very soon. But for right now, if there are numbers that are above 0, it means the source. So you're adding stuff to the atmosphere. And if numbers are below 0, it's a sink, which means you're removing stuff from the atmosphere. And in a couple of slides from now, I apologize in advance, but I'll switch it because I'll focus the lens on plants. And GPP productivity is a positive number, so some of these numbers will get flipped. But for now, what we're seeing here is that the balance of the turbulent and the storage flux reveals that carbonyl sulfide, there is a sink of carbonyl sulfide in the forest. So we can quantify how much carbonyl sulfide is taken up by the forest. And on the right, we have CO2 fluxes, which are also showing that the forest is a sink during the daytime. So this is GPP estimated from some sort of partitioning approach that also shows that the forest is a sink. So there is some sort of synergy between the diurnal cycles of these two fluxes. We also tried to study components and controls. So we looked at what was happening at the understory. So the understory is sort of, I've drawn a box around it, which is essentially what we did. Because at night time, especially when things are very calm and stable, there's no mixing. And you can really get at some processes that are happening which are, which haven't really gotten much to do with upper atmospheric mixing and all this stuff. And we saw that these patterns were, so this is again just a simple scatter plot of concentrations of carbonyl sulfide and CO2. You can see that there's a negative relationship here. And this is sort of a nighttime, so there's no photosynthesis. Increases in CO2 are happening because of respiration. At the same time, carbonyl sulfide is drawn down. So we were basically starting to get at processes other than photosynthesis that were happening, that were influencing carbonyl sulfide concentrations, which really wasn't really shown before. And I plotted these points are color coded by temperature. So at some point, at like higher temperatures, essentially this relationship starts to break down. And while respiration continues, because respiration is directly linked to temperature, carbonyl sulfide uptake stops. So we were kind of curious as to what was going on. Like I mentioned, there are many different effects in this ecosystem. So we set up these branch bags, or these chamber bags actually. So we would just put some lichen or epiphyte into a chamber and sort of measure we knew what the ambient concentration was. We measured concentrations in the chamber, tried to get at what the epiphytes are doing, what we found was, we did some measurements in situ, and what we found was that epiphytes were a sink of carbonyl sulfide. So here you have the red is CO2, blue is COS, and so this is nighttime data. We left some species of moss, I think it was, that we left in the chamber just to observe what it was doing during this transition. So this is sort of evening, sunset, night, and then sunrise, and this orange line here is incoming power, so how much light there is. So you can see that, during the night, as the epiphyte is respiring, it's also consuming carbonyl sulfide. So we needed to account for this somehow in our estimate of leaf level fixation if we were to get at photosynthesis. We also brought back these epiphytes in the lab, and we tried to sort of do these simple exercises where we would like spray water on the epiphyte, measure it, dry it, and measure it again. We found that this sort of uptake was observed in the dark, and it depended on the hydration status of the plant. So if you were to desiccate it, it was 0, everything went down to 0. But as you increase water content, epiphytes sort of took up more carbonyl sulfide, and at some point they started to not take up as much just because of diffusional constraints. There's so much water, there's nothing diffusing in. This is important because even though there's no rainfall, remember I showed in the climate data, there's no rainfall in our site. Epiphytes are known to absorb water from ambient air, and there's often dew on calm summer nights. So these epiphytes are active in the mornings and during nighttime, needs to be sort of accounted for. So just the concluding this part of the study, we saw large vertical gradients. We inferred fluxes that showed that the forest was a large sink, and we saw that OCS was consumed by epiphytes that are abundant in the ecosystem. So I'm going to, but the larger question was, how does GPP, or gross primary productivity, photosynthesis, inferred from this estimate of OCS fluxes compare with other existing approaches, right? So that was the whole rationale behind the study. To do this, we need to sort of we need to come up with an estimate of GPP based on the OCS flux. This is done through this thing called a leaf relative uptake, which can be thought of as a currency converter. So we have two different, you have OCS currency and you have a currency rate. You can get CO2 currency, which is GPP. This leaf relative uptake has been shown to vary with the CI/CA ratio, which can be inferred from isotopes data. So we can measure the stable isotope C13 in leaves, and we can come up with the ratio of intercellular to ambient CO2 concentration, relate that to LRU, plug this in, get a GPP. So first we needed to disentangle the leaf and non-leaf OCS flux from that estimate. To use this, I used a very simple model saying that, okay, if whatever OCS is being consumed if it's to do with stomatal conductance, if that is the one key variable that is driving the leaf flux, it needs to be matched with an equivalent flux of water vapor. And this has to be balanced by the gradients of those two species. We can use vapor pressure deficit, which is sort of the atmospheric demand of moisture. So it's sort of getting at, it's the driving gradient for transpiration. So if you don't have high VPD, it's common to have low transpiration. For instance, a leaf will transpire more under drier conditions than under completely humid conditions, because the VPD is low. And we can get at the leaf to air gradient of OCS by assuming that OCS inside the leaf is 0. So it's rapidly and irreversibly consumed in the leaf. And so if we say that, okay, there's no OCS in the leaf, a gradient reduces to OCS outside the leaf, which we have measurements of. So again, this is that diurnal cycle. I looked at a couple of different estimates, which is the purple and the green lines of OCS flux, which sort of were very similar. So I had some confidence in in using these numbers. I can talk about all of these different these different numbers later if you'd like. But essentially, we were able to get at an ecosystem flux, which is the yellow line, and the leaf flux, which is the green and the purple lines, which are very almost identical to each other, especially during midday. Now, as I flip the sign convention, so we can use this. We can use either the blue and the green line, plug it in here to get at GPP. We know CICA from isotopes, and then flip sign convention to confuse everyone. And we can see that photosynthesis inferred from carbonyl sulfide is higher than that from eddy covariance data. And this is shown, again, these are diurnal cycles, OCS and ECDT, which is essentially a partitioning algorithm for GPP. So the blue line is sort of much higher than the red line, which is sort of the conventional estimate. But they have a similar seasonal cycle, which is sort of, peaks in the springtime, declines as soil moisture declines. And then there's some sort of transient recovery here in September following rainfall, and then goes back again. So we know that there are many similarities, but the magnitudes are very different. So to look at which one is correct, we also estimated GPP independently from sap flow. We use measurements of sap flow in different trees, in these big trees, to sort of come up with an estimate of stand level transpiration. To do this, we use sap flow measured at the base of Douglas fir and hemlock trees. We had some sort of an idea of how much of the forest is composed of hemlocks and how much of it is composed of Douglas fir trees. And we found that most of the transpiration is from hemlock. So the shorter trees, but much more in number, contributed to most of the transpiration at the site. We can use a similar approach using CICA ratios and VPD to get at GPP from sap flow. We found that GPP sap flow was also higher than what GPP is as predicted by eddy covariance estimates. And these are two different partitioning approaches, and I can talk about those separately. But essentially, we have two independent estimates of GPP or photosynthesis at the ecosystem scale, which were higher than what the conventional methods are giving us. So we try to use a process model. So this is called SCOPE. Soil canopy observations of photosynthesis and something else it's called. But it's this process model which models photosynthesis as a function of radiative transfer. So you can feed in leaf area across the canopy and bunch of different micrometeorological conditions, radiation, energy, temperature, etc. And biochemistry, some models of stomatal conductance, etc. And so sort of like, is this somewhat powerful model that can be run at the site level by feeding in all these different data and can model GPP. So one way to check if the model was working was looking at these, again, the CICA ratios, which is very similar from observed values from isotopes. So this is sort of very characteristic CICA curve with light. Shows that CICA sort of saturates at high light conditions, and these light saturated values match observational data. It's important to note that these models sometimes lack soil moisture feedbacks, so estimates are prone to be higher because it doesn't really, if there are no soil moisture limitations in your model, then your photosynthesis flux is not constrained by that at all. We found that GPP from SCOPE was also higher from GPP from eddy covariance. Again, these are sort of seasonal cycles which show very similar seasonal patterns, but the magnitudes are off again. So putting it all together, this is, the purple line is SCOPE, which is sort of the upper end of this spectrum of what GPP at the site is. Again, it doesn't have all that soil moisture information in it, so we sort of know it to be a little higher. And then these are the two different partitioning approaches that I didn't really get to talk about much, but this is sort of conventional eddy covariance estimates, which are lower. OCS flux fits nicely in the middle, conveniently for me, but essentially, and the green are monthly means from sap flow data. So we can kind of see that all of these approaches give us an answer that is somewhat different from what we get from these partitioning approaches. And within these partitioning approaches, which is the yellow and the red, I didn't really talk about this much, but essentially there is some sort of spurious increases in this red line, which at higher temperatures, and this is somewhat a function of how the models are fitted. But essentially, this was sort of an attempt to look at GPP from a variety of different sources at one site, and we found that most of the stuff we looked at showed that photosynthesis by this forest was higher than what was predicted by these standard partitioning approaches that are employed globally at these sites. So bringing it all together, sort of ending this, we know that net ecosystem exchange is a balance of these two flows, GPP of ER. So if you have an error in estimation of one, it propagates to the other. These are chamber-based measurements of CO2 flux. We had some soil chambers. Saw that the surface measurements of CO2 flux are much higher than what the ecosystem flux from the tower was predicting. And there are issues in scaling up chamber-based estimates to ecosystem scales, but essentially showing us that there is a discrepancy in what the towers can see at night, and that propagates to daytime estimates of GPP. So in conclusion, finally, we saw that the old growth forest was a large sink of carbonyl sulfide. We found that epiphytes were a moisture-dependent sink of carbonyl sulfide. We modeled FOCS to show that canopy uptake was related to stomatal dynamics. We inferred GPP from OCS, and it was higher than what we inferred from eddy covariance approaches. And we used other approaches also. And this really highlights the value of employing multiple approaches at the same site, to look at one thing but throw everything at it to get good constraints. I think, yeah, and then finally that underestimation of ecosystem respiration likely leads to underestimation of GPP. And so showing that, you can use carbonate sulfide or sap flow or any of these other approaches to sort of correct for these underestimations. You can sort of, have a process level answer to by process level corrections to this problem of underestimation. That has been documented extensively, actually, especially from tall canopies. So just like to acknowledge co-authors, collaborators, my advisor Chris Still at Oregon State University. He's a plant ecologist, sort of ecosystem ecologist, does a lot of work on C3, C4 grasses, etc. Committee members David Noon, Max Berkelhammer at UI Chicago, Rick Meinzer at Forest Service, other collaborators, Sonia Wharton at Lawrence Livermore National Lab, Mary Whelan at UCLA and Stanford, Ken Bible, and a bunch of other people who helped me conduct this research. Finally, like to thank my sponsors, the College of Forestry at Oregon State, NASA, who funded 2 years of my studying this stuff in a forest, National Science Foundation, and the company that made the instrument and sort of made us available to it, Los Gatos Research. That's it.
OCS in the air, because I was just thinking, because sulfur concentrations have also increased globally.
Yeah.
And so whether it's likely that OCS is at these levels is a new phenomenon than say CO2, or I mean, do we know, what do we know?
We know some stuff. For instance, there was a very interesting study looking at carbonyl sulfide trapped in ice cores from Antarctica, and that was sort of so yes, carbonyl sulfide has been in the atmosphere for a long time. It's sort of amongst the reduced sulfur gases, it's the most prominent one. So that study was looking at how productivity has changed by again looking at the same sort of carbonyl sulfide CO2 ratios across different ice cores, across an ice core, let's say, at different times, looking at how productivity has changed. The other thing is, yes, there are anthropogenic emissions of carbonyl sulfide in addition to oceanic emissions, and some people have been looking at that. So, for instance, rayon manufacturing emits OCS in the atmosphere. There are emissions from fires. That's something I'm working on, different project. But yeah.
Couple of questions. Did one of your slides say that OCS and CO2 have similar pathways?
Yeah, I can go back to it.
Okay. Yeah. So I'm wondering. So is that the same for C3, C4, and CAM plants, or
Yeah,
is there variation in how it's?
Yeah, there is. So the uptake pathway is still the same. However, I talked about the leaf relative uptake LRU, which is sort of, I said it was a currency converter you could use. So that has a relationship with light, and that differs for C3 and C4 plants a little bit. So C4 plants, if I could. Yeah, so essentially LRU has this, LRU is sort of like, it's sort of a ratio, not exactly of the concentrations, but flux is normalized to the concentration. So you can plot it against light. And for C3, it sort of looks like this. For C4, it will look like that. But again, very few measurements beyond the leaf level of both these, especially C4 plants, have been done. One of my committee members, he has a site in Illinois that's looking at C3, C4. So he's looking at the same site. 1 year it was a C3 site, 1 year C4 site. But his work hasn't been published yet, so I'm not sure at larger scale what that difference looks like. But yeah.
And I think your first or second slide, you will be showing CO2 levels, global CO2 levels.
Yeah.
So what were the, are there those interannual amplitudes? Yeah, what explains that?
Yeah, so so these squiggles are interannual variations. So this is the northern hemispheric, essentially, okay, so two things. One, most of the land is concentrated in the northern hemisphere, right? So this is sort of, this drawdown is mostly because of northern hemispheric photosynthesis. That is the signal you can see here. Yeah.
I was wondering, I mean, have you guys looked at OCS soil levels? What is happening in the soil?
In where?
With OCS, I mean, we know carbon dioxide is a big, the soil is the sink of carbon dioxide. So what is the situation with OCS?
Yeah, we had some measurements also, but I didn't show them just in the interest of time. But yeah, so this whole theory of carbonyl sulfide being a one-way flux from the atmosphere into the leaf is complicated a little bit by soils, which are shown to be both they are shown to be both sources and sinks, and it's sort of dependent on soil type, but mostly on temperature and soil moisture. So very warm, dry soils are known to be a source of OCS, but cool, wet soils are often a sink. Also, the magnitude of that uptake is much smaller usually. So in my site, for instance, if you know, here peak uptake is about, peak uptake here is about 65 picomoles per meter square per second. Soil flux is peak at about 8 or 6 or something. So an order of magnitude lower often.
If there are no more questions, thank you, Bharat.
Thank you so much.
Yeah.